Okay, welcome everybody. I will be talking about how to build a primate towards authentic models of primate embryogenesis. And I would like to thank very much for the opportunity being here. There's lots of colleagues from Cambridge and beyond. And it's going to be a great pleasure talking about the most exciting highlights of what we have been up to in the lab over the last 2 years. So why looking at primates? And the reason is that, as we have already heard in the previous talk a bit, that primate embryogenesis differs quite dramatically from the rodent. And that is with regard to the what the epiblast is doing. So this is the embryonic lineage here, and this is highlighted in this cartoon of rodent development in blue, in the blues here. So you can see the epiblast, that's the lineage that forms the embryo, forms in the mouse this sort of cup shape, and we can derive stem cells from that. So epiblast stem cells and embryonic stem cells that represent two distinct pluripotent states, pre-implantation versus post-implantation. And now, if we look at the primate situation, we see something else. We have this amniotic cavity formation here, and the primates undergo this an additional lineage decision. So they form amnion, so the epiblast forms amnion, and also a flat embryonic disc. And so the questions we try to address in the lab is how do the embryonic and the extraembryonic lineages interact to make human and non-human primate embryogenesis happen? And this slide sort of summarizes our approach that we take. And this is a very simple one. And the first one is just we look what is happening in vivo, and we use the marmoset, is a small New World monkey, and we look what happens during embryogenesis and characterize embryogenesis by spatial embryo profiling. The second bit is then what we would like to do is we would like to capture these developmental stages in vitro and then put them together and assemble in vitro models for primate implantation. So we look at various systems, but to start us off, I would like to begin with our efforts on spatial embryo profiling. And because these specimens, that is the implanting primate embryo, is a very precious sample, so we want to extract really all the information we can get from this system. So we have come up with this approach for spatial embryo profiling, and the way it works is we have the marmoset uterus here, and the marmoset uterus is sectioned into many sections, and we then subject those in alternate sections to two different things, and that's RNA sequencing, and we have an imaging pipeline. And what we do is we keep track of the location of every transcriptome and then bring that together in 3D models. And I will show you a bit about that over the next couple of slides. This is what marmoset implantation looks like. This is a cross section of the uterus. You can see the endometrial glands here. This is the uterine cavity. And this slide has been stained for OCT4, our key marker for embryonic pluripotency. And this labels the very early, just after implantation, the embryonic disc here. You can also see a very thin layer. Here we've got the amniotic cavity, and this is the amnion that's sort of lining this cavity. having or originating from the epiblast. We have TFAP2C as a marker here, which is a trophoblast marker, and SOC17, which highlights to some degree the visceral endoderm. We'll come to that in a second, and the endometrial glands. Now, what the people in my lab have done is they've done lots of these sections, and we have put it together, and something I'd like to show you in this animation. We've put it together with our single cell RNA sequencing data from pre-implantation development. And this allowed us to have a consistent data set from zygote to late gastrulation, covering Canini stages 5, 6, and 7. And these are the models. There's something wrong here. Sorry about that. So what I wanted to show is the makeup, because I appreciate these structures are not that trivial. And to understand how they come from a blastocyst, I think is important. So here we have the trophoblast at the bottom, which invades into the uterus, into the uterine tissues. Then we have this layer of extraembryonic mesoderm, and the extraembryonic mesoderm then grows around the whole embryo. So this is why you have this sort of cap here, and this is at the later stages. We then have amnion and embryonic disc, which forms this bubble here. So this is the embryonic disc. This is the actual embryo here, 1, 2, and 3. And as this symposium is dedicated to germ cells, I thought I will highlight here the primordial germ cells in our Canini stage 6 embryo, which are sort of at the posterior side, in between the epiblast and the amnion. And we'll talk a bit more about that later. Then we have, on top of the embryonic disc, the visceral endoderm. And the visceral endoderm is very critical. It orchestrates gastrulation, it orchestrates embryogenesis, it patterns the embryo. It's a very important extraembryonic lineage. And what primates do is another speciality about primate development, which unfortunately we don't have time to cover today, but they form a secondary yolk sac. And we think we have some evidence to, or indications how this secondary yolk sac forms, maybe from the visceral endoderm after the first wave of primary yolk sac development. So these are our models. And what I would like to show you now is how they were made, how they were built. So what we have here is the sort of raw section, which is stained, which we then, where we then assign lineages. And here we go. And just to illustrate again, because we will need this later, this sort of the architecture. We have the trophoblast, extraembryonic mesoderm, then we have this amniotic cavity with the embryonic disc, anterior, posterior gastrulation is happening here. We have the visceral endoderm, the secondary yolk sac, and everything is covered by the extraembryonic mesoderm. PGCs in the posterior, and if we put these different sections then together, we can reconstruct in 3D these the gross morphology of the implanting primate embryo. Now, what we then wanted to do is to integrate these reconstructions with 3D transcriptomics. And very briefly take you through that. So this is what the raw data looks like. This is the cross section. The signal here is SOX2, which is a pluripotency anterior neural marker. And what you can see here are the laser microdissected samples, which are about 1 to 3 cells, is the sort of resolution we are working with here. The raw signal, the raw counts, the raw feature counts from the RNA sequencing, and you can see this sort of gradient going through here. And what we have then done is we've used a statistical model called Gaussian process regression to fill in the missing gaps, and we can sort of fill in the whole 3D space with these gene expression profiles. And the very exciting thing about this is now we have a tool to systematically find gradients, correlations, anti-correlations for all the genes in the primate embryo. This is an example here for SOX2. So this is the embryonic disc here. And we can see if we slice through this embryonic disc, we can we get a solid expression gradient. When we do this now globally, when we ask, so what are all the genes on the anterior side? What are all the genes on the posterior side? We can do spatial GOTAM analysis. We get strong features of gastrulation, differentiation in the posterior, and we get, and this was quite surprising, we see a lot of stem cell maintenance in the anterior. And what this led us then to ask is how does the embryonic niche sustain pluripotency for that long in the primate embryo? It's over 10 days in the marmoset. And so we looked into the different cross-section, different stages, signaling pathways. So what stood out to us is that so you have the different stages, 5, 6, and 7 here. And this is a cross-section through the 3D model with the gene expression plotted for the genes indicated here. So we have very high expression of nodal signaling coming onto the embryonic disc from the visceral endoderm. We see the co-receptor crypto TDGF1 in the embryonic disc here. And we see lots of BMP signaling coming from the bottom. In the extraembryonic mesoderm, amnion, we have BMP4 expression. We see downstream target ID2 there. And interestingly, noggin, the inhibitor, again coming from the top. So what this suggests to us is that this is sort of the specific embryonic niche in the primate embryo to sustain pluripotency and sort of let it exit in a controlled way. Now, BMP signaling is detrimental to pluripotency. We know that also from in vitro experiments. But as we have heard in the nice previous talk, of course, it's required for primordial germ cells. So if we look at primordial germ cells, and we find primordial germ cells in all of the 3 stages, just highlighting the one here in the Canigi stage 6. If we look at the cross, so this is the 3D model, then this is the cross section here. So we find them in the posterior end, sandwiched in between embryonic disc and amnion. And as we have heard, they express SOX17. They express TFAP2C and PLIMP1, PIDM1, but in contrast to the mouse, they're negative for SOX2. So we come then now to the amnion. If they are there in between the amnion, we want to just introduce quickly the amnion is this protective bag that forms around the embryo. You can see it here. This is a stage beyond the ones that I've shown you before, so that you see what happens with the amnion. And it forms this fluid-filled sac. And the key point here, it undergoes massive expansion. We started to look at this now at the transcriptome level. So this is a transcriptional map, how the amnion lineage emerges in the primate from the compacted morula to the ICM epiplast, and then the 2 lineages segregate. And what we find is that there's a strong similarity between those lineages, between the amnion and the embryonic disc. And so if we look at the markers here, so this would be TFAP2C is expressed in the early amnion. OCT4 is shared. OCT4, the pluripotency marker, is shared. It's lower in the amnion, but it's sort of sustained. And SOX2, consistent with the very pioneering studies of Mitinori in the cynomolgus monkey, SOX2 is downregulated very early on. But then one of the things, so how does this whole patterning of the come about? If we look at BMP signaling, we see it from the bottom coming up, but also we see this ID1, ID2, and ID3 expression of the downstream target, sort of indicating active BMP signaling in this transitional zone between where the embryonic disc and the amnion come together. And there are two sites for that, anterior and posterior in the primate embryo. And this is, I think, may explain what, why we can make amnion in vitro models quite efficiently from post-implantation cells, because that's what's happening in the embryo all along. It has to sort of constantly feed into amnion. And we're working on in vitro models to show that functionally. So this is the sort of bigger picture of what we see here. So we have nodal coming from the top and BMP from the bottom, and we have this transitional zone where we think epiblast makes embryonic makes amnion. Of course, we have also Wnt signaling in the bottom, and that is important and consistent with primordial germ cell formation. I would like to quickly come to two more points, and that is how we can use this system to use.
Out of time.
Sorry, out of time?
Time, yeah.
Okay, that's fine. Can I then jump to the acknowledgement slide, if I may? Because it's been very lucky to work with fantastic people who did all of this. So this is Sophie doing laser capture microdissection, Marquis working in the lab, Dylan works on trophoblast. This is Max, who is working on the hypoblast. Erin works on pluripotency, and she's very swift with her pipette. We have a collaboration with Shrinjan on super resolution. Erika Sasaki has provided embryos. This is Stan, who works on the mathematical modeling. Clara did some of the embryoids, and this is Chris, who did the Gaussian process regression. Anna works on microfluidics, and Oriol, who is discovering the amnioids here, and Timo, who doesn't like to be filmed. And with that, I'd like to thank you. I'm happy to take questions.